All right, welcome to the nervous system, fast. So the nervous system in the human body is composed of both the central nervous system and the peripheral nervous system. The central nervous system, or the CNS, is the brain and the spinal cord. So don't forget that spinal cord when you think of the central nervous system. And then the peripheral nervous system, or all the other neurons in our bodies. The nervous system is made of two main types of cells, glia and neurons. Right? So first we're going to talk about glia, which are really the neuronal support cells for the body. In the central nervous system, we have microglia, these little kind of starry shaped ones. They are like macrophages, and they are scavengers that phagocytose and remove dead cell and debris. Oligodendrocytes are in the central nervous system as well, and they kind of wrap around these neurons, as you can see here in blue, and they produce myelin in the central nervous system. And that wrapping really helps the neurons fire faster and more efficiently. We also have Schwann cells. These are in the peripheral nervous system, and they wrap around axons as well to help conduct signals faster. When we look at neurons, we have three main types. We have sensory neurons. These are the ones in the peripheral nervous system, sensing your environment. Then we have interneurons, which are in the central nervous system only, and they connect neurons to one another. And then we have motor neurons, which go from the central nervous system out to the peripheral nervous system to cause stimulation of muscle contractions, organ function, and glands, so releasing of some of those hormones that we learned about in the endocrine system, right? Going into the central nervous system is afferent, so sensory are afferent, and then the motor neurons are efferent, so the efferent, and I always remember that because they're causing an effect, right? I just want to show you one quick example of one of those circuits. So here's a neuronal circuit that you guys are all familiar with. It's called the knee-jerk reflex. So here we have sensory neurons in the patellar region of the knee. They send an excitatory signal here in blue. They're sensing that, so you have an afferent response going into the spinal cord. You have small interneurons in that spinal cord region, and then you have motor neurons that are going out to the hamstring and the quadricep, causing stimulation of those muscles and that knee-jerk response that we're all aware of. I want to point out that there's no brain involvement at all, and that's why this can happen without you really even realizing it. So this is one of those reflexes, goes to the central nervous system, but no brain involvement at all. So let's dive into what neurons look like. So the cell body of a neuron is a typical eukaryotic cell with all of the cell organelles and functions that you normally have learned of. For example, the endomembrane system, mitochondria, lysosomes, all the good stuff. On that cell body, you have these long, elaborate processes that are reaching out, and those are called dendrites. Those are on the cell body, and they're really important for receiving information from all over the cell's environment. The axon is this long cellular extension that helps transmit information from the dendrite out to the end of the cell. At the end here, we have axon terminals. These are often called synaptic terminals, and they send out information. So information comes in at the dendrite, they come out at the axon. Out at the synaptic terminal. The axons are wrapped with what we heard of before, glial cells, right? So an example, this is peripheral nervous system, and we are looking at the myelin-covered axon. In myelin are glial cells that wrap around the axons to help neurons fire. So if we look at a nerve synapse, or the place where one neuron is sending information out and another neuron is receiving that information. That's called the nerve synapse. So here is the presynaptic cell, which would be this one, which is sending the information. And here we have a postsynaptic cell, which is going to be receiving that information. Just like any cell signaling that we know of, we have a signal being sent out, which is in the form of a neurotransmitter here in blue. It's going to be released into this synaptic cleft, which is a very, very, very short distance. And the postsynaptic cell must have the appropriate receptors for that neurotransmitter in order to have a cellular response. Neurons and neuron. So neuron to neuron, this is a neuron to neuron connection. And one thing that's cool about neurons is that they really convert chemical information in the form of neurotransmitters into electrical signals, which are going down these axons through an action potential, right? So electrical signal stimulating a chemical change, which would then signal another electrical change. So if we look at what a normal resting neuron cell looks like, it has what's called a membrane potential of negative 70 millivolts, right? So again, whenever we're looking at a cell or a picture like this, you need to orient yourself on what's inside and what's outside, right? So we know about the sodium-potassium pump. We know that sodium is going to go outside and potassium is going to go inside with the use of ATP. When we look at that, the function of the sodium-potassium pump causes a membrane potential. And what that means is it's forcing a lot of sodium out and it's putting a lot of potassium in. And what that causes is that the membrane potential is going to be minus 70, right? And what that means is that it's a difference in charge across the plasma membrane. The inside is more negative than the outside. And you can look here. These are both +1. We're shoving 3 out, and we're only putting 2 in. That is, by definition, going to make the membrane, the inside of the cell, more negative. And there's some good videos if you want to watch about that. Gated ion channels are really important for neuronal function. So just a reminder, we have what are called voltage-gated ion channels. And how these function is that they are open if the membrane potential changes. So these are driven by an electrical charge. So here, on a closed state, we have a voltage-gated ion channel, and we also have an extracellular ligand-gated ion channel, right? So if the membrane potential is negative 70, nothing happens. However, when there is a change in that membrane potential, what can happen is that the voltage-gated ion channel can open. Remember, ligand-gated ion channels only open if another protein or chemical binds to them, right? So this is voltage-gated. These are ligand-gated. Okay, so let's talk about an action potential going, again, from the cell body down this very long axon to that terminal, which the whole point of a action potential is to release neurotransmitters in the form of at that synaptic cleft. So electrical signal is going to travel down the axon. Here are the kind of here are the steps, and we're going to look again at this graph. We're looking at voltage on the y-axis and time on the x-axis. Remember, the voltage of a resting neuron cell is negative 70 millivolts. If we get a stimulation of the neuron, if it's not past a threshold of negative 55 millivolts, nothing's going to happen, right? But if we do get past that threshold, what we see is called a depolarization, where sodium channels open, and because there's a concentration gradient with sodium more outside, sodium's going to rush inside of the cell, causing the inside of the cell voltage to go very, very positive, right? That's the first step of the action potential. Once it reaches a very high point, then the sodium channels close and the potassium channels open. And remember, based on that sodium-potassium pump, there's a lot of potassium in the cell, meaning when those channels open, so potassium is going to rush out. And it's going to rush out and continue to leave until the cell gets what's called hyperpolarized, meaning even lower than -70. And then finally, potassium channels are going to close, and the sodium-potassium pump is going to restore that resting state. In axons that have myelin, these electrical currents, these action potentials, go through called saltatory conduction, where action potentials jump to only the regions that are not covered by myelin, and those are called the nodes of Ranvier. All right, these are really nice animations if you want to see about action potential again. And I just want to focus again on the nerve synapse. So again, the nerve synapse here, we're looking at a presynaptic cell sending an electrical current down right at this synapse. This synapse is then going to have a chemical response. It's going to release protein or another molecule onto the next cell. So these responses can be excitatory, which means it's going to cause an action potential in this next cell. They can also be inhibitory, meaning they are going to suppress an action potential, making it harder to fire. And because cells have many, many dendrites, they have a lot of inputs, and they're constantly kind of managing all of those inputs to see whether this cell is then going to be going to release or inhibit its neurotransmitter release. When you look at a brain, I don't know why I have this here, but the white matter is all, I'm pretty sure that is all myelin based. All right, so maybe that was my segue into the human brain. So the human brain has three kind of parts to it. The very first, which is the most evolutionarily ancient, is called we call it the lizard brain. This is the brain stem and the cerebellum, right? These are involved in all the daily functions, kind of homeostasis, life and death roles, and in particular the fight or flight response, which we know can be controlled by a few hormones like epinephrine. Then we have what is called the mammalian brain, and we call them mammalian brain because in mammals, this is one of the evolutionary hallmarks, right? We went from kind of this lizardy brain into the mammalian brain. This is called the limbic system. This controls emotions, memories, and habits. It also helps in decision making. And then the human brain is a really complex evolutionary adaptation called the neocortex. And this is involved in all of those extra things like language, abstract thinking, imagination, consciousness, and it's involved in reasoning and rationalization. And again, tissues of the brain, white matter is myelinated with those glial cells, meaning they're really conductive and fast. And then we have the gray matter, just because you may have heard of those terms before. The nervous system is really important for controlling two main things. The first is called the sympathetic nervous system, or the sympathetic responses. This is stimulation throughout the body, and it prepares the body for a fight or flight response. So again, when you have epinephrine signaling, and that comes from your adrenal glands. I might have said that wrong in the last one. So if it comes from your adrenal glands on top of your kidney, and that will stimulate all of these things throughout your body, ready to make you fight or flight. The parasympathetic system is the opposite. It relaxes the body. You get the opposite effect. So the parasympathetic system is really kind of triggered when you do things like mindfulness and meditation, which is really good and restorative. So the nervous system can also have dysfunction, and a lot of these neurological or neurodegenerative diseases that are probably going to affect some of you at some point in your life. So Alzheimer's disease is something that many of us have already heard of. It is neurodegeneration in that cortex region of the brain, and this is really it has really detrimental effects on memory, and in particular short-term memory. Then we have ALS, which is also called Lou Gehrig's disease, and this is progressive degeneration of specifically motor neurons. So these are the neurons that have the effect on muscles, glands, and organs. Parkinson's disease is neurodegeneration of neurons that are really important for body movement, and Parkinson therefore has some very characteristic body phenotypes. Multiple sclerosis is another one where we get destruction specifically of the myelin in the central nervous system. And again, this is an autoimmune. So your own immune system starts recognizing this myelin as foreign and starts attacking it.